Shroud of Turin.
Yeah.
And I always thought that was fake.
And they found that it was like 2,000 plus years old.
Scientists have no idea how they faked it. It's got to be real.
The DNA results loaded on Professor Gianni Barcaccia's screen at 11:47 p.m. For 30 seconds, nobody in the University of Padua Genetics lab said a word. Then someone whispered, "That's not possible." The sequencing software had mapped genetic origins for the particles extracted from the Turin Shroud. But instead of pointing to one region, medieval France, where skeptics claimed it was forged, the data pointed everywhere. China, India, Africa, the Middle East, Europe, ancient DNA from every corner of the known world, pressed into a single piece of cloth. The team ran the analysis again, same result. A third time, same result. One researcher stepped outside to catch his breath. Another just kept shaking his head. Because what they were looking at didn't just challenge the forgery theory. It mapped an ancient journey that historians had dismissed as legend for centuries. And that was before they found the pollen, before they analyzed the blood, before they discovered what the 1988 carbon dating had gotten catastrophically wrong. The DNA was just the beginning. For centuries, the Shroud of Turin has divided the world. Believers call it the authentic burial cloth of Jesus Christ, a silent witness to the resurrection. Skeptics call it the most sophisticated medieval forgery ever created. Some have blamed Leonardo da Vinci himself. But here's the deal. In 2015, a team of geneticists did something unprecedented. They didn't just examine the shroud, they interrogated it at the molecular level. And what they found didn't just challenge existing theories, it systematically demolished them. This is the story of how modern science cracked open secrets hidden for 2,000 years. And why do those secrets make some people very uncomfortable? The photograph that changed everything. But before we get to what that DNA revealed, you need to understand what made scientists take this cloth seriously in the first place. On May 28, 1898, an Italian lawyer named Secondo Pia hauled camera equipment the size of a suitcase into Turin Cathedral. He'd been granted rare permission to photograph the shroud during a public exhibition. Photography back then was brutal. Slow exposures, magnesium flashes, glass plates. Late that night, alone in his darkroom, Pia lowered a photographic plate into developing chemicals. As the image emerged, he nearly dropped the glass. Think about that for a second. This man was a seasoned photographer. He knew exactly what to expect from a negative. Light becomes dark, dark becomes light. Simple. But on the shroud's negative, a perfect positive image appeared. A face, deep-set eyes, a broken nose, a beard, bruising on the cheek. For the first time in history, the hidden figure became visible. And it looked like a photograph of a real man. Here's the catch. For an image to appear this way on a photographic negative, the original would have to already be a negative, meaning someone created a perfect reverse tone. Image 800 years before photography existed. No medieval artist could have done this. The human eye cannot perceive the world in negative tones. There was no way to preview the result, no way to verify it. The skeptics had their first problem, but they didn't know yet how bad things were about to get. The DNA extraction. For over a century, researchers examined the shroud with every tool available. X-rays, ultraviolet light, infrared imaging, laser scanning. But the real secrets weren't in the image itself. They were hiding in the dust. Professor Barcaccia's team used sterile micro-vacuum devices fitted with ultrafine filters. They collected particles not just from the surface, but from deep within the weave, spaces where ancient traces can remain sealed for millennia. Every precaution was taken. A single sneeze, a flake of skin from a technician, could contaminate samples and destroy ancient signals. The material was transported to ultra-clean laboratories. Sequencing began using next-generation technology, focusing on mitochondrial DNA, the type that survives longest in ancient material and serves as a reliable geographic marker. For weeks, computers processed millions of nucleotide sequences, comparing them against global genomic databases. And then came that night in the lab, the night that changed everything. Results that broke the theory. The findings were published in Nature Scientific Reports, and they detonated like a bomb in the scientific community. Here's what everyone expected. If the shroud was a medieval forgery made in France, as skeptics had insisted for decades,
Ever since the shroud first really appears in history, there's been controversy about it, and there have been questions about its authenticity.
European DNA should have dominated overwhelmingly. French, Italian, maybe some German. Case closed. If it was an authentic relic from Jerusalem that never traveled, Middle Eastern markers should have appeared almost exclusively. Neither happened. The shroud carried genetic traces from the Druze, an isolated ethno-religious community in the mountains of Israel, Jordan, Lebanon, and Syria. Druze DNA is extremely ancient and stable, having changed little over thousands of years. That's a powerful Middle Eastern marker. Western European haplogroups appeared too, expected, given the shroud's documented European history since the 15th century. But here's where things got strange. Haplogroups from North and East Africa emerged. Egypt, Ethiopia, regions home to some of the world's earliest Christian communities. Then genetic markers from the Indian subcontinent showed up. And get this, traces from East Asia, DNA associated with China, India, Africa, Europe, the Middle East, all pressed into one piece of cloth. The skeptics' forgery theory had just developed a fatal wound. Because here's the deal. If someone faked this shroud in a French abbey around 1350, how exactly did Chinese and Indian DNA get onto it? Medieval Europe had no contact with those regions on any scale that could leave genetic traces like this. But the DNA was only half the story. What they found in the pollen would make the genetics look like a warm-up. The pollen that shouldn't exist. Two scientists approached this mystery independently. Professor Avinoam Danin, a renowned Israeli botanist from Hebrew University, and Swiss forensic scientist Max Frey, a pioneer of forensic pollen analysis. They examined pollen grains trapped deep within the linen's fibers. Using adhesive tapes, they lifted microscopic samples from the fabric. What they found caused a sensation. 58 plant species, 17 native to Europe, exactly what you'd expect from 6 centuries of European custody. But the majority came from elsewhere. Plants native to the Middle East, Turkey, the Anatolian steppe, and then the discovery that made the room go silent. Plants that grow nowhere else on Earth except a narrow corridor between Jerusalem and Jericho. At the center was Gundelia tournefortii, a thorny desert thistle. Its pollen accounted for nearly half of all samples on the shroud. Nearly half. Think about that for a second. Why would pollen from a thorny plant dominate a burial cloth? The answer hit researchers like a freight train. The crown of thorns. Gundelia tournefortii has long, needle-like spines, exactly what Roman soldiers could have twisted into a mocking crown. And this thistle blooms around Jerusalem in early spring, during Passover. Basically, someone would have to believe that a medieval French forger traveled to Israel, collected microscopic pollen from plants growing only between Jerusalem and Jericho, transported it back to Europe without contamination, and applied it invisibly to a cloth, all to fool scientists who wouldn't exist for another 600 years. That claim collapsed under its own absurdity. Pollen cannot be painted. It acts as an invisible seal from the crime scene, a geographic fingerprint that cannot be forged. But the botanical evidence was about to be overshadowed, because when researchers finally analyzed the blood, what they found made everything else look tame. Blood that screams. For years, skeptics insisted the reddish marks on the shroud were pigments. Ochre, cinnabar, tempera mixed with gelatin, the work of a clever artist. In 2017, that claim was destroyed. Italian researchers examined the stains using transmission electron, microscopy, and Raman spectroscopy. At the nanoscale, they found no pigment. They found blood. Human blood. Type AB, one of the rarest blood types on Earth. It's not that simple, though. This wasn't the blood of a healthy person. Within the samples, scientists identified nanoparticles of creatinine and ferritin bound to hemoglobin. And here's where the implications terrified the research team. Such extreme concentrations of creatinine appear only under one condition: severe fatal trauma, prolonged torture, massive muscle destruction. When tissue is destroyed by repeated blows, a process called rhabdomyolysis floods the bloodstream with creatinine. The blood on this cloth recorded exactly that. One researcher described it as a biochemical scream of pain. And get this, the bloodstains remain red. Normally, ancient blood turns dark brown or black over centuries, but analysis revealed unusually high levels of bilirubin, a substance released by the liver during extreme physical stress. Bilirubin preserves the red coloration indefinitely. The blood of a tortured man stays red. This isn't mysticism, it's biochemistry. An artist can paint a wound. No artist can fake the biochemical signature of a body in hypovolemic shock. But one massive obstacle still stood in the way. The 1988 radiocarbon dating that supposedly proved the shroud was medieval. That result had dominated headlines for 30 years. It was about to be demolished. The 1988 disaster. In 1988, 3 prestigious laboratories announced their results with 95% confidence. Oxford, Zurich, Arizona. The fabric dated to between 1260 and 1390. Medieval, a forgery. The world accepted the verdict. Even the church stepped back. But here's the catch. Science never stops. And 30 years later, researchers found the catastrophic error. The sample used in 1988 was cut from the very edge of the shroud. The corner most handled over centuries, grasped by bishops, touched by pilgrims, exposed to candle smoke, sweat, and skin oils. But it gets worse. Chemist Ray Rogers of Los Alamos National Laboratory discovered something devastating. That corner had been expertly repaired during the Middle Ages. Cotton threads had been woven in and dyed to match the original linen. The repair was so skillful, it went undetected for centuries. The 1988 laboratories hadn't dated the shroud. They'd dated the medieval patch. They'd measured the age of cotton fibers and accumulated grime, not first-century linen. This wasn't a minor oversight. It was a catastrophic sampling error, like determining the age of an ancient statue by testing the gum stuck to its base. In 2022, Italian physicist Liberato De Caro applied a completely different approach, wide-angle X-ray scattering. This technique measures the aging of cellulose at the atomic level, bypassing surface contamination entirely. Over time, the polymer chains in linen naturally degrade. This acts as the material's internal clock. De Caro compared the shroud against fabrics of known ages, from Egyptian mummy wrappings to medieval textiles. The results stunned everyone. The shroud's molecular structure didn't match medieval linen at all. It was far older. It matched with remarkable precision linen fragments recovered from the fortress of Masada in Israel. Masada fell in 74 AD. Those textiles are dated between 50 and 74 AD, the first century, the time of Christ. The 1988 results hadn't just been wrong, it had been wrong for over 1,000 years. The image that defies physics. One mystery remains, and it's the one that keeps physicists awake at night. How did the image form? There are no brushstrokes, no pigments, no ink. The image exists only 200 nanometers deep on the fiber surface, hundreds of times thinner than a human hair. Scrape it, and it vanishes. It's not paint. It's a chemical transformation caused by oxidation and dehydration. Basically, a burn left by an unknown energy source. Scientists tried everything: acid, heat, gamma rays. Only one thing could replicate it: a short, intense pulse of vacuum ultraviolet radiation. Here's the problem. Such lasers didn't exist in antiquity. They barely exist now. To form the image across 4 square meters, the body would have had to emit an unimaginable burst of energy, lasting less than a billionth of a second. Enough to imprint the cloth, not enough to destroy it. In 1976, NASA technology revealed something even more disturbing. The shroud contains an anatomically accurate 3-dimensional image. The intensity of the imprint corresponds exactly to the distance between the body and the cloth. No artist, ancient or modern, could reproduce this. It's not how art works. It's not how forgery works. Layer by layer, discipline by discipline, biology, chemistry, physics, geology, the evidence converges on one location and one time frame: Jerusalem, somewhere between 30 and 33 AD. The question they can't answer. The shroud has been returned to its vault in Turin. It rests in silence, but the data doesn't rest. It keeps scientists arguing in conferences, publishing papers, running new analyses. Because no matter how many times they test it, the same impossible results keep appearing. So, here's the question that keeps researchers awake at night. If this cloth recorded a burst of energy that our best technology cannot replicate, if it captured DNA from an ancient journey that skeptics said never happened, if the blood contains biochemical markers of torture that no artist could fake, what exactly happened in that tomb, too? 1,000 years ago? And why are some people still so desperate to prove it never did?